All right, everyone, welcome to week 3. This week we're shifting gears slightly in our hematology journey. Up to this point, we've been focused primarily on hematopoiesis, how blood cells develop in the bone marrow. You learned about stem cells, differentiation pathways, and the progression from immature precursor cells to mature circulating cells. Now we're going to move out of the bone marrow and into the peripheral blood, which is where most clinical hematology interpretation actually happens. Our focus this week is on circulating leukocytes, or white blood cells, and more specifically, three things. First, how to recognize them morphologically on a smear. Second, what their functions are in the immune system. And third, how normal variation differs from reactive changes, which is where hematology starts becoming really interesting. As histotechnologists, even though you may not be performing differentials every day, you will absolutely encounter hematopoietic tissues, bone marrow biopsies, lymphoid organs, inflammatory infiltrates, and understanding what these cells are and what they're doing gives you a much deeper understanding of what you're seeing in tissue sections. So this week is really about building your hematology eyes. We're going to move from simply recognizing a cell to asking two questions. What cell am I looking at? And maybe even more importantly, what is that cell doing? So let's talk about the transition we're making this week. Last week we focused on hematopoiesis and maturation, essentially the developmental journey that blood takes inside the bone marrow. We looked at how hematopoietic stem cells differentiate into the various lineages and how those cells mature from immature precursor forms into functional cells. That maturation process follows very predictable patterns. Cells change in size, nuclear chromatin structure, cytoplasmic composition, and granule development as they mature. Those predictable changes are what allow laboratory professionals to recognize normal maturation and eventually abnormal development. But once cells finish maturing in the marrow, they don't just stay there. They enter the circulatory system, and that's where we start to see them on a peripheral blood smear. Now, the focus changes slightly. Instead of asking what stage of maturation is this cell, we start asking what type of mature cell is this? And once we identify the cell type, we ask another important question. What role does this cell play in the immune system? Because every leukocyte you see on a smear is there for a reason. The distribution of cells you observe can tell you a lot about what the body may be responding to, as an infection, inflammation, allergic reactions, or other physiologic stressors. So this week we're moving from development to interpretation. Let's briefly revisit where we left off. In week 2, we spent a lot of time talking about hematopoiesis and cell maturation within the bone marrow. That environment is where blood cells are produced, differentiated, and prepared to enter circulation. Inside the marrow, you'll have a highly regulated process where hematopoietic stem cells generate progenitor cells, which then develop into the various mature blood cell types, like red blood cells, platelets, and the different leukocyte lineages. The key concept from last week was that cells progress from immature to mature forms, and during that progression, their morphology changes in predictable ways. Now, once those cells finish their maturation process, they move out of the marrow and into the peripheral circulation. That's what we're focusing on now. We're no longer looking at immature precursor cells. Instead, we're looking at fully differentiated cells performing their immune functions in the bloodstream. So instead of identifying a myeloblast versus a promyelocyte, we're identifying things like neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And in the laboratory, when we examine a peripheral blood smear, we're essentially seeing a snapshot of what the immune system is doing at that moment in time. That distribution of cells can provide valuable clues about the body's physiologic state. Now you might be wondering, why do we care so much about a peripheral blood smear? Peripheral blood is actually an incredibly useful diagnostic window into what's happening throughout the body. First, it reflects immune system activity. If the body is responding to a bacterial infection, a viral infection, a parasitic infection, or an allergic response, those immune cells will often change in number or appearance. Second, peripheral blood can show physiologic stress. Trauma, inflammation, infection, and systemic disease can all influence the types of leukocytes circulating in the bloodstream. Third, the peripheral smear is often one of the earliest clues of systemic disease. Before more complex testing is performed, changes in cell morphology or distribution can alert clinicians that something abnormal may be happening. And perhaps one of the biggest reasons peripheral blood smears are so widely used is that they are quick and relatively inexpensive. A simple smear can provide a remarkable amount of diagnostic information. One of my favorite ways to think about this, and you'll probably hear me say this more than once, is this. A blood smear is basically a snapshot of what the immune system is doing right now. Every cell you see on that slide is there for a reason, and our job as laboratory professionals is to learn how to interpret what that picture is telling us. All right, now we're getting into one of the most important practical skills in hematology, evaluating a peripheral blood smear. A peripheral smear is not meant to replace automated testing like the CBC. Instead, it complements it. Think of it this way. The CBC gives us the numbers, but the smear tells us what those numbers actually look like. So whenever we evaluate a smear, we always correlate it with the CBC results. If the CBC shows an elevated white blood cell count, the smear helps us determine which cells are elevated and why. If the CBC flags abnormal indices, the smear lets us look at the actual morphology of those cells. There are three major things we evaluate when examining a peripheral blood film. First is red blood cell morphology. We look at cell size, shape, color, and variation. This is where we identify things like anisocytosis or poikilocytosis, hypochromia, or abnormal inclusions. Second is the white blood cell differential, which is the process of identifying and counting the different leukocyte types present in circulation. Third is the platelet estimate. Platelets are tiny and sometimes clump, so automated counters can occasionally misinterpret them. A smear allows us to visually confirm whether platelet numbers appear adequate. Another major goal of smear evaluation is identifying abnormal cell distribution or unusual morphology. That might include immature cells appearing in circulation, reactive changes in leukocytes, or abnormal red cell forms. Rodak emphasizes this concept quite a bit. The peripheral smear acts as a visual verification tool. Automated analyzers are incredibly useful, but morphology is still one of the most powerful diagnostic clues we have. So in short, CBC provides the data, the smear provides the context. Now let's talk about one of the foundational concepts in hematology, the white blood cell differential. Conceptually, the differential count is pretty straightforward. Traditionally, it involves examining a peripheral blood smear and counting 100 leukocytes, identifying each one as you go. By the end of that process, you calculate the percentage distribution of each cell type: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So if you counted 100 cells and 60 of them were neutrophils, then neutrophils represent 60% of the differential. But here's where students sometimes get tripped up. The differential gives us relative percentages, not actual cell numbers. To understand what's really happening clinically, we often convert those percentages into absolute counts using the total white blood cell, the WBC from the CBC. For example, a patient might have 80% neutrophils. That sounds high, but if the total white blood cell count is low, the absolute neutrophil count might actually be normal or even decreased. So percentages alone can be misleading without the CBC context. Another important thing to remember is that neutrophils are the most common circulating leukocyte, typically making up about 50 to 70% of circulating white cells in healthy adults. That's why when you perform a manual differential, you'll feel like you're seeing neutrophils constantly, because biologically they dominate the circulation. One practical tip I always give students is this: Always know the normal distribution before deciding something is increased. You can't recognize abnormal patterns unless you understand what normal actually looks like. All right, now we move into the skill that every hematology student eventually develops, identifying leukocytes on a smear. And if you're new to this, it can feel overwhelming at first. You're staring at a microscope slide thinking, these all look like purple blobs. That feeling is completely normal. The key to identifying leukocytes isn't memorizing dozens of pictures. It's learning to systematically evaluate morphologic features. Experienced technologists don't just glance at a cell and magically know what it is. They subconsciously walk through a series of morphologic clues. Things like, what is the size of the cell? What does the nucleus look like? How condensed is the chromatin? What color is the cytoplasm? And are there granules present? When you evaluate cells in a consistent order like that, identification becomes much easier. Another helpful mindset is remembering that leukocytes aren't random shapes. They're highly specialized immune cells, and their morphology reflects their function. For example, neutrophils have segmented nuclei because they need flexibility to migrate through tissues. Monocytes have abundant cytoplasm because they're precursors to macrophages that engulf debris. Lymphocytes have dense nuclei because they are primarily focused on immune signaling. So when you start thinking about function alongside morphology, leukocyte identification starts making a lot more sense. Now let's break down a step-by-step strategy for identifying leukocytes on a peripheral smear. And here's the most important rule, the one I repeat constantly in the lab. Size first. Always size first. Before you look at anything else, determine how large the cell is relative to nearby red blood cells. Red cells provide a built-in reference scale on every smear. Once you have a sense of size, move on to the nuclear shape. Is the nucleus round, dented, segmented into multiple lobes? Nuclear shape is one of the strongest distinguishing features between leukocyte types. Next, examine the chromatin pattern. Is it coarse and clumped or fine and delicate? Mature lymphocytes, for example, tend to have very dense chromatin, while immature cells have more open chromatin. Then evaluate the cytoplasm. Look at the color. Is it pale, light blue, grayish, or deeply basophilic? Finally, determine whether granules are present, and if they are, what they look like. Granules can vary in size, staining characteristics, and abundance depending on the cell type. This stepwise method works because it mirrors the way cells actually mature biologically. As cells develop, they typically become smaller, chromatin condenses, nucleoli disappear, cytoplasmic color changes, and granules appear in certain lineages. So in many ways, identifying leukocytes is really about recognizing where a cell sits along that maturation spectrum. And once you get comfortable using this systematic approach, leukocyte identification becomes much less intimidating and honestly kind of fun. All right, now that we've talked about starting with cell size, we need a reliable reference point. And fortunately, nature gives us one built directly into every blood smear, the red blood cell. Red blood cells are remarkably consistent in size. In a healthy individual, most red blood cells measure about 7 to 8 micrometers in diameter. That makes them a perfect internal measuring stick whenever you're evaluating a smear. So when you're looking at leukocytes under the microscope, you should always be mentally comparing them to nearby RBCs. For example, if you see a leukocyte with a nucleus that is roughly the same size as a red blood cell, you're probably looking at a small lymphocyte. That's one of the classic visual anchors students learn early in hematology. At the other end of the spectrum, monocytes are typically the largest normal leukocytes in circulation. When you see a cell that's clearly larger than surrounding red blood cells, often 2 to 3 times their diameter, monocytes should immediately come to mind as a possibility. Neutrophils and eosinophils generally fall somewhere in between, larger than red blood cells but not nearly as large as monocytes. The key takeaway here is that visual comparison is essential. Hematology isn't about memorizing exact measurements in micrometers. It's about recognizing proportional differences. Experienced technologists don't think in numbers when they're looking at a smear. They're constantly asking, how big is this cell compared to the red cells around it? And once you get comfortable using RBCs as your reference anchor, identifying leukocytes becomes much more intuitive. Once you've established cell size, the next major clue we evaluate is the nucleus, specifically the chromatin pattern. Chromatin structure tells us two important things: cell type and cell maturity. In fact, one of the fastest ways to recognize leukocytes is by learning their characteristic nuclear patterns. Let's start with mature lymphocytes. These cells have very dense, clumped chromatin, often described as looking like a soccer ball or blocky pattern under the microscope. The nucleus takes up most of the cell, leaving only a thin rim of cytoplasm. Next are neutrophils, which have their signature segmented nuclei. Mature neutrophils typically have 3 to 5 nuclear lobes, connected by thin strands of chromatin. This segmentation gives neutrophils flexibility, allowing them to migrate through tissues during inflammation. Monocytes look quite different. Their nuclei are often described as folded, indented, or lacy. Sometimes they appear kidney-shaped or horseshoe-shaped. The chromatin is looser than in lymphocytes, giving the nucleus a softer appearance. Eosinophils, on the other hand, usually have bilobed nuclei, often resembling a pair of sunglasses. But the nucleus is actually less noticeable because their bright cytoplasmic granules tend to steal the show. A helpful phrase I like to remind students of is this: chromatin tells you maturity. As cells mature, chromatin becomes more condensed and clumped, reflecting decreased transcriptional activity. Immature cells, like blasts, have much more open and delicate chromatin. So whenever you're identifying leukocytes, take a moment to study the nucleus. It's often the most reliable clue you'll have. After evaluating the nucleus, the next place we look is the cytoplasm. Cytoplasmic features often help confirm what the nucleus already suggested. Each leukocyte type has characteristic cytoplasmic staining patterns and granule features. Let's start with neutrophils. Their cytoplasm is typically a pale pink or neutral color, which is actually where the name neutrophil comes from. The granules are small and fine, and they often blend into the cytoplasm unless you look closely. Lymphocytes, on the other hand, usually have sky blue cytoplasm, and typically very little of it. In small lymphocytes especially, the nucleus dominates the cell, leaving only a thin rim of cytoplasm. Monocytes have some of the most distinctive cytoplasm in the leukocyte family. It's often described as gray-blue, and it frequently contains vacuoles or irregular textures. That appearance reflects the cell's function as a precursor to macrophages, which are highly active phagocytic cells. Then we have eosinophils, which are probably the easiest leukocyte to recognize because of their large, bright orange-red granules. These granules contain proteins involved in parasitic defense and allergic responses. Finally, basophils contain dark purple, coarse granules that can sometimes obscure the nucleus entirely. These granules contain histamine and other inflammatory mediators involved in hypersensitivity reactions. So when you're evaluating cytoplasm, you're looking at three things: color, granules, and texture. Together with nuclear features, these cytoplasmic clues help you narrow down the identity of the cell. Now that we've established the identification framework, size, nucleus, chromatin, cytoplasm, and granules, we're ready to start examining specific leukocyte types. And we're going to begin with the most common circulating white blood cell, the neutrophil. In healthy adults, neutrophils typically account for about 50 to 70% of circulating leukocytes, making them the dominant player in the peripheral blood smear. Neutrophils are the first responders of the immune system. Their primary job is to respond quickly to bacterial infections inflammation, and tissue injury. When bacteria invade tissues, neutrophils are among the first immune cells recruited to the site. They migrate out of the bloodstream, move through tissue spaces, and begin engulfing pathogens through a process called phagocytosis. Inside the neutrophil, specialized granules contain enzymes and antimicrobial substances that help destroy bacteria and cellular debris. One interesting thing about neutrophils is their relatively short lifespan. Once released from the bone marrow, they circulate for only about 6 to 10 hours before migrating into tissues. After performing their defensive roles, they often undergo apoptosis or contribute to the formation of pus during infection. In laboratory medicine, neutrophils provide valuable clues about inflammatory processes. Changes in neutrophil numbers or morphology can signal infection, stress responses, or bone marrow activity. So as we move forward, we'll look more closely at neutrophil morphology, normal variations, and reactive changes that can occur during immune responses. All right, let's take a closer look at neutrophils, the workhorses of the circulating immune system. As we mentioned earlier, neutrophils are the most abundant white blood cell in peripheral blood, typically making up about 50 to 70% of the differential count in healthy adults. So when we're scanning a smear and performing a differential, we're going to see a lot of neutrophils, and that's exactly what you should expect. Biologically, neutrophils are, again, the first responders of the innate immune system. When bacteria invades tissues, neutrophils are among the very first immune cells recruited to the site of the infection. They respond rapidly, sometimes arriving within minutes of inflammatory signaling. One of the reasons neutrophils are so effective at responding quickly is that the bone marrow maintains a very large reserve pool of mature neutrophils ready for release into circulation when needed. In fact, the bone marrow contains significantly more neutrophils than are actually circulating at any given time. Another interesting feature of neutrophils is their relatively short lifespan. Again, once released into circulation, they typically live only 1 to 3 days before undergoing programmed cell death. That short lifespan means the body must constantly produce new neutrophils, which is why bone marrow activity is so tightly linked to neutrophil count. Functionally, neutrophils perform several important tasks. They phagocytose bacteria, release antimicrobial enzymes from their granules, and can even form structures called neutrophil extracellular traps, or NETs. These DNA-based structures trap pathogens and help prevent them from spreading through tissues. And if you've ever wondered what pus actually is, a large component of it is dead neutrophils that have completed their defensive mission. So in many ways, neutrophils are like the frontline infantry of the immune system. They're fast, aggressive, and very disposable. Now let's focus on what a mature neutrophil actually looks like under the microscope. The defining feature of a mature neutrophil is its segmented nucleus. Typically, the nucleus contains 2 to 5 lobes connected by very thin strands of chromatin. Those thin connections are sometimes called nuclear filaments. This segmented structure is not just decorative. It actually helps neutrophils move through tissue. The flexible nucleus allows them to squeeze through endothelial gaps during diapesis, the process where immune cells exit the bloodstream and migrate into inflamed tissues. Another important feature is the cytoplasm, which tends to appear pale pink or neutral in color. This is why neutrophils are called neutrophils again. Their cytoplasmic granules stain relatively neutrally compared to other granulocytes. Inside the cytoplasm are fine, barely visible granules. These granules contain enzymes such as lysozyme, defensins, and myeloperoxidase, which play key roles in destroying bacteria and other pathogens once they've been engulfed. When identifying neutrophils on a smear, the combination of segmented nucleus, pale cytoplasm, and fine granules is usually very distinctive. One thing to keep in mind is that neutrophil segmentation increases with maturity, but there is a normal range, typically 2 to 5 lobes. When you start seeing significantly more lobes than that, it may indicate certain clinical conditions, which we'll talk about later in the course. So when you're scanning a smear and you see a cell with a multi-lobed nucleus and subtle granules, chances are very good you're looking at a neutrophil. Now let's connect neutrophil morphology with function, because understanding what these cells actually do makes their structure a lot more meaningful. One of the primary roles of neutrophils is phagocytosis, which means engulfing and digesting microorganisms or cellular debris. Once a neutrophil encounters bacteria or damaged tissue, it essentially surrounds the target and internalizes it into a structure called a phagosome. Inside that phagosome, neutrophils release antimicrobial substances stored in their cytoplasmic granules. This process is called degranulation, and it delivers enzymes and antimicrobial proteins directly to the pathogen. Neutrophils also produce reactive oxygen species, sometimes abbreviated as ROS. These molecules help destroy bacteria, but can also contribute to local tissue damage during inflammation. Another fascinating function of neutrophils is something called netosis. During this process, neutrophils release strands of their own DNA into the extracellular environment, creating web-like structures called neutrophil extracellular traps. These traps help immobilize bacteria and prevent them from spreading through tissues. All of these processes contribute to acute inflammation, which is the body's immediate response to infection or injury. Now, here's a helpful tip for smear interpretation. When looking at neutrophils, the number of nuclear lobes does not tell you how activated the cell is. It primarily reflects maturity. Students sometimes assume that more lobes means a more aggressive neutrophil, but in reality, segmentation mostly reflects how long the cell has been maturing. So remember, segmentation equals maturity, not activity. Now we're going to talk about a slightly less mature form of the neutrophil called the band neutrophil, sometimes simply referred to as a band. Band neutrophils are essentially young neutrophils that have not yet developed a fully segmented nucleus. Instead of multiple distinct lobes, the nucleus appears as a horseshoe-shaped or curved band of chromatin. The key thing to remember is that bands do not have distinct nuclear segmentation. If you can draw clear separations between nuclear lobes, you're looking at a segmented neutrophil. If the nucleus is one continuous curved structure, it's likely a band. Bands are normally present in small numbers in circulation because the bone marrow releases a small percentage of slightly immature cells as part of normal hematopoiesis. However, when the body is dealing with a significant infection or inflammatory stress, the bone marrow may release neutrophils more quickly than usual. When this happens, we start to see increased numbers of band neutrophils in circulation. This phenomenon is referred to as a left shift. The term comes from the way early hematology charts arranged neutrophil maturation stages from left to right. Immature cells on the left, mature cells on the right. So a left shift essentially means the bloodstream is showing more immature neutrophil forms than usual, often indicating that the bone marrow is responding to infection or inflammation. Recognizing bands correctly is an important skill during differential counts because it can provide valuable clues about the body's current immune response. All right, now we're shifting gears a little bit and moving into our next major white blood cell group, the lymphocytes. Up to this point, we spent time talking about neutrophils, which are part of the innate immune system, the body's rapid, nonspecific response to infection. Neutrophils show up quickly, attack aggressively, and don't really care what the pathogen is as long as it doesn't belong there. Lymphocytes, on the other hand, are part of the adaptive immune system. This is the portion of immunity that is specific, targeted, and capable of memory. In other words, lymphocytes are a little more strategic than neutrophils. Instead of just attacking everything immediately, they recognize specific antigens and mount a coordinated immune response. From a hematology perspective, lymphocytes are the second most common white blood cell type in peripheral blood, and they play a huge role in protecting the body against viral infections, abnormal cells, and long-term immune threats. Morphologically, lymphocytes look very different from neutrophils. Instead of a segmented nucleus in abundant cytoplasm, lymphocytes are typically small, round cells dominated by their nucleus. And that difference is something you'll start to recognize very quickly when reviewing peripheral blood smears. So in this section, we're going to look at the basic characteristics of lymphocytes, how to recognize them on a smear, the different functional types of lymphocytes, and how their structure supports their immune role. Because in hematology, just like in histology, structure almost always gives you clues about function. Lymphocytes typically make up about 20 to 40% of the white blood cell differential in healthy adults. So when you're performing a differential count, lymphocytes are usually the second most common cell you'll encounter after neutrophils. But while they may look fairly similar morphologically on a blood smear, lymphocytes actually represent several different functional cell types. The three main categories you'll hear about are B lymphocytes, T lymphocytes, and natural killer cells, or NK cells. Each of these plays a slightly different role in immune defense. B cells are responsible for producing antibodies, which are proteins that recognize and bind specific antigens. Once an antibody binds a pathogen, it helps the immune system neutralize or eliminate it. T cells are involved in cell-mediated immunity. Some T cells coordinate immune responses by signaling other immune cells, while others directly attack infected or abnormal cells. And then we have NK cells, which function a little differently. They are technically lymphocytes, but behave more like part of the innate immune system. NK cells can recognize and destroy virus-infected or tumor cells without prior antigen exposure. Now here's the tricky part for students learning hematology. You usually cannot distinguish B cells, T cells, and NK cells based solely on morphology in a routine blood smear. They look very similar under the microscope. Differentiating them typically requires immunophenotyping techniques such as flow cytometry, which identify surface markers on the cell. So when you're performing a manual differential, you'll usually classify these cells simply as lymphocytes, even though functionally they may represent several different immune cell types. Now let's talk about what a typical small mature lymphocyte looks like on a peripheral blood smear. The most striking feature of this cell is its large round nucleus. In fact, the nucleus occupies most of the cell's volume, which is why we describe lymphocytes as having a high nuclear to cytoplasmic ratio, or high NC ratio. The chromatin inside the nucleus tends to appear dense and clumped, giving it a very dark purple appearance with bright Giemsa staining. Surrounding the nucleus is usually just a thin rim of pale blue cytoplasm. Sometimes that cytoplasm is so thin that it can be difficult to see unless you're looking closely. So visually, lymphocytes often look like a dark purple circle with just a little halo of cytoplasm around it. Here's a helpful practical tip when scanning the smear. If the cell looks like it's almost all nucleus, your first thought should probably be lymphocyte. Another point to keep in mind is that lymphocytes are typically about the same size as red blood cells, or sometimes just slightly larger. That size comparison is actually really helpful when you're differentiating cells during a manual count. If you see a cell that's round, small, very dark, and roughly RBC sized, that's very likely a small mature lymphocyte. And once you start doing more smear reviews, your brain will start recognizing that pattern almost automatically. Now let's connect the structure of lymphocytes to their function, because that relationship actually makes a lot of biological sense. As we mentioned earlier, lymphocytes play central roles in adaptive immunity. B lymphocytes produce antibodies that bind specific pathogens. When activated, B cells can differentiate into plasma cells, which are essentially antibody-producing factories. T lymphocytes come in several functional varieties. Helper T cells coordinate immune responses by releasing signaling molecules called cytokines, which activate other immune cells. Cytotoxic T cells directly attack infected or abnormal cells by inducing apoptosis. There are also regulatory T cells, which help keep immune responses balanced so the system doesn't become overactive. And finally, natural killer cells, specialized in recognizing cells that appear abnormal, particularly virus-infected cells or tumor cells, and destroying them without needing prior sensitization. Now, if we think back to the morphology we just discussed, the large nucleus of lymphocytes reflects their role in genetic regulation and immune signaling. These cells rely heavily on gene expression and protein synthesis to produce cytokines, antibodies, and other signaling molecules. So while neutrophils are built for rapid attack and destruction, lymphocytes are built more for communication, coordination, and targeted defense. Different strategies, different cell designs. And as you continue studying hematology and pathology, you'll see how changes in lymphocyte populations can tell us a lot about viral infections, immune disorders, and hematologic diseases. All right, now we're moving into the next leukocyte category, monocytes. Up to this point, we've talked about neutrophils, which are rapid responders in acute inflammation, and lymphocytes, which are responsible for targeted immune responses and immune memory. Monocytes fall somewhere in between those two worlds. They are part of the mononuclear phagocyte system, and they play a major role in cleanup, antigen presentation, and long-term immune coordination. One important thing to understand about monocytes is that what we see circulating in the bloodstream is actually just a transitional stage in their life cycle. Monocytes circulate in the blood for a relatively short period of time, and then they migrate into tissues where they differentiate into macrophages or dendritic cells. Once they enter tissues, these cells become powerful phagocytes that help remove pathogens, cellular debris, and damaged tissue. In histotechnology and pathology, this is particularly relevant because macrophages are commonly seen in tissue sections, especially in areas of inflammation, chronic infection, or tissue injury. So when you hear the term monocyte in blood, you can think of it as the circulating precursor to macrophages in tissue. And in peripheral blood smears, monocytes are relatively easy to recognize once you know what to look for, mainly because they tend to be big, a little irregular, and a little messy looking compared to other white cells. So in this section, we're going to talk about what monocytes are, what they look like on a smear, and why they matter clinically and in tissue pathology. Monocytes typically make up about 2 to 8% of the white blood cell differential, so they are less common than neutrophils or lymphocytes, but still an important part of the immune system. One thing that makes monocytes stand out immediately on a blood smear is their size. Monocytes are usually the largest circulating white blood cells. When you're scanning a smear, they often appear noticeably larger than surrounding red blood cells and larger than most lymphocytes. Another key point is that monocytes are the precursors to macrophages and dendritic cells. Once monocytes leave the bloodstream and enter tissues, they differentiate into these more specialized cells that perform a variety of immune functions. Macrophages are essentially the body's professional phagocytes. They engulf pathogens, remove dead cells, and help coordinate immune responses. Dendritic cells, on the other hand, are extremely important for antigen presentation. They capture antigens and present them to leukocytes, helping activate the adaptive immune response. So monocytes play a really important bridging role between innate and adaptive immunity. From a laboratory standpoint, increased numbers of monocytes, something called monocytosis, can sometimes be associated with chronic infections, inflammatory conditions, autoimmune disorders, or certain hematologic diseases. So when you see elevated monocytes on a CBC differential, it often suggests that the body is dealing with longer-term immune activity rather than an immediate acute response. Now let's look at what monocytes actually look like on a peripheral blood smear, because their morphology is pretty distinctive once you get used to it. The first thing most people notice is the nucleus, which is typically kidney-shaped, folded, or sometimes horseshoe-like. Unlike the very dense chromatin seen in lymphocytes, monocyte chromatin tends to appear looser and more delicate, sometimes described as lacy or reticular chromatin. The cytoplasm of monocytes is usually abundant and gray-blue in color. This grayish appearance is sometimes referred to as ground glass cytoplasm. Another feature you may see in monocytes is cytoplasmic vacuoles, which are small, clear spaces within the cytoplasm. These vacuoles can form as part of phagocytic activity. Monocytes also tend to have irregular cell borders, which contributes to that slightly messy appearance compared to the neat round shape of lymphocytes. Here's a practical smear reading tip that many hematology students find helpful. If the cell looks large, gray, and a little messy, your brain should immediately start thinking monocyte. Another way to think about it is this. Neutrophils equals segmented and organized. Lymphocytes equals small and mostly nucleus. Monocytes equals big, gray, and slightly chaotic. And once you start scanning enough blood smears, those visual patterns becoming surprisingly easy to recognize. Now that we've talked about what monocytes look like, let's talk about what they actually do, because function is really where these cells shine. One of the primary roles of monocytes is phagocytosis. Like neutrophils, monocytes can engulf bacteria, debris, and damaged tissue. However, monocytes tend to be more involved in longer-term cleanup and immune coordination rather than the immediate rapid attack we see with neutrophils. Once monocytes leave the bloodstream and enter tissues, they differentiate into macrophages, which become some of the body's most effective phagocytic cells. Macrophages can ingest pathogens, remove dead cells, and help clear inflammatory debris during tissue repair. Another critical function of monocytes in macrophages is antigen presentation. After engulfing a pathogen, these cells process pieces of that pathogen and present them on their surface to T lymphocytes. This interaction helps activate the adaptive immune response. So monocytes are really acting as a kind of bridge between innate and adaptive immunity. Monocytes also play a role in chronic inflammation and wound healing. Macrophages release signaling molecules called cytokines that regulate inflammation and help recruit other immune cells to areas of tissue damage. From a pathology standpoint, macrophages are extremely important because they appear in many chronic inflammatory conditions, granulomas, and areas of tissue injury. So if neutrophils are the first responders, macrophages, which originate from monocytes, are often the cleanup crew and long-term managers of the immune response. Now we're moving into one of the most visually recognizable white blood cells, the eosinophils. If neutrophils are subtle and monocytes are messy, eosinophils are basically the flashy cells of the leukocyte world. These cells belong to the granulocyte family, meaning they contain prominent cytoplasmic granules that stain strongly with Wright Giemsa stain. Eosinophils get their name from the dye eosin, which stains their granules a bright red-orange color. So when you're scanning a blood smear and suddenly see a cell that looks like it's filled with tiny sunset-colored granules, your brain should immediately start thinking eosinophil. Functionally, eosinophils play important roles in parasitic infections, particularly infections caused by helminths, which are parasitic worms. They're also heavily involved in allergic reactions and hypersensitivity responses, including conditions like asthma, allergic rhinitis, and eczema. Because of that, increased numbers of eosinophils, called eosinophilia, can often be seen in patients with allergies, parasitic infections, or certain inflammatory conditions. From a smear interpretation standpoint, eosinophils are actually one of the easiest leukocytes to identify, mainly because their granules are so distinctive. And once you see a few of them under the microscope, they tend to become pretty memorable. Eosinophils are relatively uncommon compared to other white blood cells. Normally, they make up about 1 to 4% of the white blood cell differential. Even though they're less common, they are extremely important in specific immune responses. As we mentioned earlier, eosinophils are particularly effective against parasitic infections, especially larger parasites that can't easily be phagocytosed by other immune cells. Instead of engulfing parasites, eosinophils release toxic proteins from their granules, which help damage or kill the parasite. Some of the substances released from eosinophil granules include major basic protein and eosinophil peroxidase, which are very potent antimicrobial molecules. Eosinophils are also heavily involved in allergic and inflammatory responses. In conditions like asthma, eosinophils can accumulate in tissues and contribute to airway inflammation. Another interesting aspect of eosinophils is their lifespan and migration pattern. They typically circulate in the blood for only a short period of time, roughly 8 to 12 hours, before migrating into tissues where they may remain for several days. So in many ways, the bloodstream is just a temporary transit system for eosinophils before they reach their target tissues. Clinically, elevated eosinophil counts can give important clues about allergic disease, parasitic infection, or certain immune disorders. Now let's talk about what eosinophils look like under the microscope because their morphology is extremely distinctive, as I've mentioned. The nucleus of an eosinophil is typically bilobed, meaning it has two nuclear lobes connected by a thin chromatin strand. Some students describe it as looking like a pair of sunglasses or a dumbbell shape. But the most obvious feature of eosinophils is their cytoplasmic granules. These granules are usually large, uniform, and intensely red-orange when stained with right Giemsa stain. They are much more prominent than the fine granules seen in neutrophils. These granules contain the antimicrobial proteins that allow eosinophils to attack parasites and participate in inflammatory responses. Another important point is that the granules are usually fairly uniform in size and distribution, which helps distinguish eosinophils from other granulocytes. Here's a simple visual tip that many hematology students remember. If the cell looks like it belongs in a sunset painting, it's probably an eosinophil. Bright red-orange granules plus a bilobed nucleus is the classic eosinophil look. And once you've seen a few eosinophils on a smear, they become one of the easiest leukocytes to identify during a differential count. Now that we've talked about what eosinophils look like, let's talk about what they actually do, because their granules contain some pretty powerful substances. One of the most important components of eosinophil granules is major basic protein, which is highly toxic to parasites. When eosinophils encounter large parasites, especially helminths or parasitic worms, they can't easily engulf them the way neutrophils engulf bacteria. So instead, eosinophils attach to the parasite and release their granule contents in a process called degranulation. Those toxic proteins damage the parasite's surface and help kill or weaken it. Eosinophils also play a major role in modulating inflammation. They release signaling molecules that influence other immune cells and help regulate the inflammatory response. This is particularly important in allergic reactions where eosinophils often accumulate in tissues like the lungs or skin. For example, in asthma, eosinophils contribute to airway inflammation and tissue irritation. That's why some asthma treatments specifically target eosinophilic inflammation. From a clinical lab perspective, eosinophilia, an increased eosinophil count, can give us important diagnostic clues. It often points towards allergic disease, parasitic inflammation, or certain inflammatory conditions. So even though eosinophils aren't very common in the differential, when they increase, they often tell us something meaningful is happening immunologically. Now we arrive at the rarest granulocyte in the bloodstream, the basophil. Basophils are a kind of the mysterious introverts of the leukocyte world. They're present in extremely small numbers, and even experienced laboratorians might only see a few during a differential count. Basophils belong to the granulocyte family, just like neutrophils and eosinophils, but their granules stain deep purple to dark blue with the right Giemsa stain. These granules contain important inflammatory mediators such as histamine and heparin. Histamine is a chemical that promotes vasodilation and increased vascular permeability, which are classic features of allergic reactions. Heparin, on the other hand, has anticoagulant properties, helping regulate clotting and localized inflammatory responses. Basophils are most closely associated with hypersensitivity reactions, particularly allergic responses. In many ways, they function similarly to mast cells, which are related immune cells found in tissues rather than circulating in the blood. So although basophils are rare, they play a very important role in allergy, inflammation, and immune signaling. Basophils are actually the least common white blood cell in the peripheral blood. Normally, they make up less than 1% of the total white blood cell differential, and sometimes you may not see any in a routine differential count. Because they're so rare, students often have trouble identifying them at first, but once you understand their morphology, they become easier to recognize. Functionally, basophils are heavily involved in hypersensitivity reactions and allergic responses. When activated, basophils release substances like histamine, leukotrienes, and cytokines that promote inflammation and recruit other immune cells. This contributes to the classic symptoms we associate with allergic reactions, things like swelling, redness, itching, and increased mucus production. Basophils can also be elevated in certain myeloproliferative disorders, such as chronic myelogenous leukemia. So although they are rare in healthy blood, persistent basophilia can sometimes be an important diagnostic clue in hematologic disease. In short, basophils may be small in number, but they have powerful biochemical effects when activated. Now let's talk about what basophils look like on a peripheral blood smear, because their appearance is pretty distinctive. The most notable feature of a basophil is its coarse, dark purple granules. These granules are usually large, irregular, and densely packed, and they often stain such a deep purple color that they can partially or completely obscure the nucleus. The nucleus itself is typically bilobed or S-shaped, but in many cases you can barely see it because the granules are so prominent. This is actually one of the best identification clues. If you're scanning a smear and you see a cell that looks like someone spilled dark purple ink all over it, that's a strong hint you may be looking at a basophil. Another feature is that basophils often have a somewhat irregular cell outline, again due to the dense granulation. So here's a simple practical tip for smear interpretation. If the granules are so dark and dense that the nucleus is hard to see, think basophil. And because they're so rare, spotting one during a differential often feels like a little victory for laboratory scientists. Now that we've walked through all of the major white blood cell types, it's helpful to step back and compare them side by side. In clinical hematology, recognizing the differences between leukocytes is critical for performing accurate manual differentials and smear reviews. Each leukocyte has its own combination of features, things like nuclear shape, granule appearance, cytoplasm characteristics, and relative abundance in the blood. For example, neutrophils typically have segmented nuclei and fine pale granules, and they make up the majority of circulating white blood cells. Eosinophils stand out because of their bright red-orange granules and bilobed nuclei, making them relatively easy to identify. Basophils, on the other hand, contain dense, dark purple granules that often obscure the nucleus, and they're the rarest leukocytes in the differential. Monocytes are the largest leukocytes, with abundant gray-blue cytoplasm and often a kidney-shaped nucleus. And lymphocytes usually have a round nucleus with a thin rim of cytoplasm and are heavily involved in adaptive immunity. Learning to distinguish these cells quickly is a key skill for anyone working in hematology, pathology, or clinical laboratory science. And like most microscopy skills, the real secret is simple: practice, practice, and more practice. The more smears you review, the faster your brain starts recognizing these patterns automatically. Before I move on from this slide, I want to take a second to share with you a mnemonic saying that I kind of use to identify the descending order of how these leukocytes appear in the blood. So it is never let monkeys eat bananas. So never stands for neutrophils, which are the most abundant. Let is the lymphocytes. Monkeys, monocytes. Eat, eosinophils. And bananas is the basophils. Now let's look at an important distinction you'll see in hematology in differential counts, the difference between a band neutrophil and a segmented neutrophil. Both of these cells belong to the same lineage and both are neutrophils, but the difference lies in maturity. A segmented neutrophil is the fully mature form. Its nucleus is divided into multiple distinct lobes, usually between 2 and 5, connected by thin chromatin strands. This segmentation is a hallmark of a mature neutrophil and reflects its final stage of development. A band neutrophil, on the other hand, is slightly less mature. Instead of distinct lobes, the nucleus appears as a curved band or horseshoe shape without clear segmentation. Think of it as a nucleus that hasn't quite finished folding yet. In clinical practice, the presence of increased band neutrophils is referred to as a left shift. This typically occurs when the bone marrow is responding to infection or inflammation and releases neutrophils into circulation a little earlier than usual. For example, during bacterial infections, the body ramps up neutrophil production and may push more immature forms, like bands, into the bloodstream. From a laboratory perspective, recognizing the difference between bands and segmented neutrophils is important during manual differentials because it can provide clues about the patient's immune response. And when you're looking at smears, remember this quick tip. If the nucleus forms separate lobes, it's segmented. If the nucleus looks like one continuous curved band, it's probably a band neutrophil. Now let's compare two cells that students often mix up when they first start looking at blood smears, lymphocytes and monocytes. Both of these cells are part of the agranulocyte group, meaning they lack the obvious cytoplasmic granules seen in neutrophils, eosinophils, and basophils. But there are several key features that help distinguish them. First is cell size. Lymphocytes are generally smaller, often close to the size of a red blood cell. Monocytes are the largest circulating white blood cells and usually appear noticeably bigger on a smear. Second is the nucleus. Lymphocytes typically have a round, dense nucleus that takes up most of the cell. There is usually just a thin rim of cytoplasm around it. Monocytes have a very different nuclear shape. Their nucleus often looks kidney-shaped, folded, or horseshoe-shaped, and the chromatin tends to be less condensed than what we see in lymphocytes. Another helpful feature is the cytoplasm. Monocyte cytoplasm is usually pale gray-blue and more abundant, sometimes with a slightly ground glass appearance. Lymphocyte cytoplasm is much thinner and more uniform. So when you're looking at a smear, ask yourself 3 quick questions. How big is the cell? What shape is the nucleus? And how much cytoplasm do I see? Those clues usually point you in the right direction. Now let's compare two granulocytes that students sometimes confuse, eosinophils and basophils. Both cells contain large granules, but their appearance is very different once you know what to look for. Eosinophils are famous for their bright red-orange granules. These granules stain strongly with eosin dye, which is actually where the cell gets its name. The nucleus of an eosinophil is typically bilobed, and the granules are usually uniform in size and evenly distributed throughout the cytoplasm. Basophils, on the other hand, look dramatically different. Their granules are dark purple to deep blue, and they are often so dense that they obscure the nucleus. That's one of the easiest ways to tell them apart. Another difference is nuclear visibility. In eosinophils, you can actually see the bilobed nucleus clearly between the granules. In basophils, the nucleus is often partially hidden or completely masked by the granules. Functionally, the two cells are also different. Eosinophils are involved in parasitic defense and allergic inflammation, while basophils play a key role in hypersensitivity reactions and histamine release. So here's a quick mental shortcut that helps a lot of students. Orange sunset granules means eosinophil. Dark purple ink blot granules means basophil. Once you recognize those color patterns, distinguishing them becomes much easier. And this final chart brings everything together and shows the major leukocyte types side by side. From a laboratory medicine perspective, this is essentially what your brain learns to do automatically when you perform manual differentials or review peripheral blood smears. First, we divide the white blood cells into two major categories, granulocytes and agranulocytes. Granulocytes include neutrophils, eosinophils, and basophils. These cells contain visible cytoplasmic granules and typically play roles in innate immunity and inflammatory responses. Neutrophils are the most abundant, making up about 60 to 70% of circulating white blood cells, and they serve as the body's primary bacterial defense cells. Eosinophils are much less common, around 2 to 4%, and are particularly involved in parasitic defense and allergic inflammation. Basophils are the rarest, usually around 1% or less, and participate in hypersensitivity reactions and histamine release. Then we have the agranulocytes, monocytes and lymphocytes. Monocytes make up about 5% of circulating white blood cells and eventually differentiate into macrophages and tissue. Lymphocytes typically account for 20 to 40% of the differential and play central roles in adaptive immunity, including antibody production and cell-mediated immune responses. From a smear reading standpoint, your brain is really looking for a few key features: nuclear shape, granule appearance, cell size, and cytoplasmic characteristics. And once you start seeing enough smears, those patterns become surprisingly easy to recognize. Or as I like to tell students, eventually your brain starts doing hematology pattern recognition mode, and you can spot these cells almost instantly. To this point in the lecture, we've focused on what normal leukocytes look like. But in real laboratory practice, blood cells do not always stay perfectly textbook. The immune system is constantly responding to infections, inflammation, stress, and other physiologic signals. When that happens, white blood cells can undergo reactive changes, meaning their appearance shifts as part of a normal physiologic response. This is important because not every abnormal-looking cell indicates disease like leukemia. Many morphologic changes simply reflect the body doing its job, responding to infection or inflammation. For example, neutrophils can develop features called toxic changes during bacterial infections. Lymphocytes may appear larger and more irregular during viral infections. Even monocytes may increase in number during chronic inflammatory processes. From a laboratory medicine perspective, the key skill is learning to distinguish between reactive changes and malignant changes. Reactive changes are typically temporary and reversible, whereas malignant changes tend to be persistent and associated with uncontrolled cell growth. So part of interpreting a peripheral smear involves asking a critical question: is what I'm seeing simply the immune system reacting to something, or is it something more concerning? That's exactly what we'll start exploring in the next few slides. So what exactly do we mean when we say reactive change? A reactive change refers to a morphologic alteration in blood cells that occurs as part of a normal physiologic response to stress, infection, inflammation, or immune stimulation. In other words, the cells may look different under the microscope, but the process itself is not malignant and not inherently dangerous. These changes happen because the bone marrow and immune system are actively responding to signals from the body. For example, during infection, the bone marrow may accelerate white blood cell production. Cells may enter circulation earlier in their maturation process, or they may develop structural features that help them function more effectively during immune responses. Another important point is that reactive changes are often reversible. Once the underlying condition, such as infection or inflammation, resolves, the blood cell morphology typically returns to normal. From a laboratory standpoint, recognizing reactive changes helps prevent overinterpretation of normal immune responses as pathologic disease. So while the cells may look different than what you see in textbooks, the key question is always, does this change reflect physiology or does it suggest pathology? That distinction is one of the core skills of hematology. One of the most important examples of reactive change involves neutrophils, particularly during bacterial infections. When the body is fighting a significant infection, neutrophils can develop several characteristic morphologic changes, collectively referred to as toxic changes. The first is toxic granulation. These are large, dark cytoplasmic granules that appear more prominent than the fine granules we normally see in neutrophils. They reflect increased enzyme production as neutrophils ramp up their antimicrobial activity. Next are Dole bodies. These appear as small, pale blue inclusions in the cytoplasm, usually near the cell membrane. They represent remnants of rough endoplasmic reticulum and are often seen during inflammatory states. Another feature you may see is cytoplasmic vacuolization, which looks like clear holes or bubbles within the cytoplasm. These vacuoles can represent active phagocytosis or cellular stress. Together, these features often indicate significant bacterial infection, sepsis, or severe inflammation. For laboratory professionals, reviewing a peripheral smear, identifying toxic changes can provide an early clue that the patient's immune system is actively responding to infection. So when you see neutrophils that look a little rougher, darker, and more busy than usual, that's often the immune system working overtime. Another classic example of reactive change occurs with lymphocytes, particularly during viral infections. Reactive lymphocytes are sometimes called atypical lymphocytes, although the term reactive is usually preferred because it emphasizes that this is a normal immune response rather than a malignant process. Compared to normal lymphocytes, reactive lymphocytes tend to be larger and more irregular in appearance. They often have more abundant cytoplasm, which may appear light blue and sometimes skirt around nearby red blood cells on the smear. Their borders may look irregular or scalloped, and the nucleus can appear slightly less condensed than in typical resting lymphocytes. These changes occur because lymphocytes are becoming activated immune cells, responding to viral antigens and preparing to carry out immune functions. Reactive lymphocytes are commonly seen in infections such as infectious mononucleosis, cytomegalovirus, and other viral illnesses. From a smear interpretation standpoint, recognizing reactive lymphocytes is important because they can sometimes look dramatic under the microscope. But remember, dramatic does not necessarily mean dangerous. Sometimes it just means the immune system is doing exactly what it's supposed to do. Now let's look at another morphologic change that laboratory professionals pay close attention to: hypersegmented neutrophils. A normal neutrophil nucleus typically has 3 to 5 lobes, connected by thin strands of chromatin. When we begin seeing neutrophils with more than 5 nuclear lobes, we refer to them as hypersegmented neutrophils. This finding is most commonly associated with megaloblastic processes, particularly deficiencies in vitamin B12 or folate. So why does this happen? Vitamin B12 and folate are essential for DNA synthesis. When DNA production is impaired, nuclear maturation slows down while the cytoplasm continues developing normally. This leads to cells with abnormally mature or overly segmented nuclei. In hematology, hypersegmented neutrophils can actually be one of the earliest morphologic clues that a patient may have megaloblastic anemia. You may detect them on a peripheral smear before other laboratory findings become obvious. From a laboratory interpretation standpoint, this is another great example of how small morphologic details can reflect underlying biochemical processes happening in the body. A careful technologist or pathologist reviewing the smear might recognize this pattern and trigger further testing for nutritional deficiencies. So again, morphology is not just about identification. It often provides the first clue to systemic disease. Before we move forward, this slide highlights one of the most important principles in laboratory medicine. Reactive changes do not automatically mean malignancy. Cells can appear unusual or dramatic under the microscope simply because the immune system is responding to infection, inflammation, physiologic stress, or metabolic imbalance. This is why clinical context matters so much. Laboratory professionals never interpret morphology in isolation. Instead, we consider it alongside other information such as patient symptoms, clinical history, complete blood count results, differential counts, and additional laboratory findings. A reactive lymphocyte in a patient with fever and viral symptoms likely represents an immune response, but a population of abnormal lymphocytes in a patient with persistent leukocytosis and lymphadenopathy might raise suspicion for a hematologic malignancy. The same cell type can mean very different things depending on the clinical picture. So a good rule of thumb in laboratory medicine is this: morphology without clinical correlation is incomplete. In other words, the microscope gives us important clues, but the full story always comes from combining laboratory findings with clinical information. Now we're going to shift our thinking slightly from what cells look like to why they look that way. In hematology, morphology often reflects cellular function. Let's break down a few examples. First, granules. Granules inside leukocytes contain enzymes, antimicrobial proteins, and other molecules involved in immune defense. When we see prominent granules, we are often observing cells that are actively engaged in enzymatic or antimicrobial activity. Next is nuclear segmentation. Neutrophils have segmented nuclei because it provides greater flexibility as the cell moves through blood vessels and migrates into tissues during inflammation. A segmented nucleus allows the cell to squeeze through tight spaces, which is critical for immune surveillance and response. Finally, we can consider the nuclear to cytoplasmic ratio, or NC ratio. Cells with a high NC ratio often have large nuclei relative to their cytoplasm. This typically indicates active transcription and protein synthesis, which is common in immature or rapidly dividing cells. So morphology is not random. Every structural feature we observe reflects something about the cell's biologic role or developmental stage. Understanding these relationships helps laboratory professionals move beyond simple recognition and toward true interpretation of what the cells are doing. This slide brings together a key concept that connects everything we've discussed so far. When we examine blood cells under the microscope, we are not just identifying structures, we are actually observing biologic activity in real time. Granules indicate chemical weaponry that cells use to destroy pathogens. Nuclear segmentation reflects mobility and flexibility, allowing neutrophils to move from circulation into tissues where infection or inflammation is occurring. Changes in nuclear size or chromatin patterns can signal cellular activation, maturation stage, or metabolic activity. In other words, morphology provides a window into cellular physiology. For laboratory professionals, this connection between structure and function is incredibly important. Recognizing a reactive lymphocyte, a hypersegmented neutrophil, or toxic granulation isn't just about labeling the cell correctly. It's about understanding what the immune system is doing and why those changes are occurring. This is what transforms hematology from simple cell identification into a powerful diagnostic tool. When we learn to link morphology to function, we move from asking, "What is this cell?" to asking the more important question, "What is this cell telling us about what is happening in the patient's body?" And that's the mindset that defines strong laboratory professionals. Now let's bring some of these concepts together and think about common physiologic patterns that laboratory professionals encounter. Different types of infections tend to stimulate different leukocyte responses, and these patterns can provide helpful clues during interpretation. For example, bacterial infections often lead to neutrophilia, meaning an increased number of neutrophils in circulation. Because neutrophils are the body's primary responders to bacterial invasion, the bone marrow increases their production and release. In many cases, you may also see reactive changes, such as toxic granulation or a mild left shift. In contrast, viral infections are more commonly associated with lymphocytosis, which is an increase in circulating lymphocytes. These lymphocytes may appear reactive or atypical, showing larger size and more abundant cytoplasm as they become activated in response to viral antigens. Finally, parasitic infections frequently stimulate eosinophilia, meaning elevated eosinophil counts. Eosinophils contain specialized granules that are particularly effective in responding to parasitic organisms and certain allergic responses. Recognizing these physiologic patterns allows laboratory professionals to begin forming interpretive connections between the blood smear and what may be occurring clinically. It doesn't provide a diagnosis on its own, but it can certainly help guide the clinical picture. One of the most important skills in hematology is learning to interpret the complete blood count together with the peripheral blood smear. Neither one tells the full story by itself. The CBC provides quantitative data, the actual numbers. For example, it tells us whether the white blood cell count is elevated, decreased, or within normal limits. But the CBC cannot show us how the cells actually look. That's where the peripheral smear becomes incredibly valuable. When we examine the smear under the microscope, we evaluate morphology, the structure, maturity, and appearance of individual cells. We may detect reactive changes, abnormal maturation patterns, or unusual cell types that automated instruments cannot always identify correctly. Another key concept here is that absolute counts matter. A differential percentage may look normal, but if the total white blood cell count is high or low, the absolute number of each cell type can still be abnormal. So the best interpretation always comes from combining the CBC values, the differential count, and the peripheral smear morphology. A diagnosis made without reviewing the smear can often be incomplete or misleading. That's why careful smear review remains such an important skill in laboratory medicine. And that brings us to the end of this section. Throughout the lecture, we've moved beyond simply identifying leukocytes and started thinking more like laboratory professionals who interpret what they see. We discussed how normal leukocyte morphology reflects underlying cellular function and how reactive changes can occur when the immune system responds to infection, inflammation, or physiologic stress. We also looked at important morphologic clues, including toxic neutrophil changes, reactive lymphocytes, and hypersegmented neutrophils, and how these findings can point towards specific physiologic processes. One of the biggest takeaways from this lecture is that morphology rarely exists in isolation. It must always be interpreted alongside clinical context and laboratory data, including the CBC and differential counts. When we combine quantitative laboratory values with careful microscopic evaluation, we gain a much clearer picture of what is happening inside the patient's body. And that's really the goal of hematology and laboratory medicine, connecting what we see under the microscope to what the body is actually experiencing physiologically. So the next time you look at a peripheral smear, remember, you're not just looking at cells, you're looking at the immune system in action.